This tutorial shows you how to do a spatial analysis of molecular variance, also known as SAMOVA, for landscape genetic analysis. This assumes you are using Ubuntu Linux version 20.04 as your operating system. If you do not have Linux, you can emulate it on your Windows or Mac computer for free. See the show notes for details. In fact, I am emulating Linux right now on my Mac computer using VirtualBox. Speaking of which, I have a VirtualBox virtual machine pre-configured with the software I will show you today, as well as other population genetics and ecological niche modeling software. See the show notes below for step-by-step instructions to download and hook up this virtual machine within your Windows or Mac computer. This tutorial assumes you have Wine installed on your Linux machine, which allows you to run Windows programs. See the link below, or you can just use the pre-configured Ubuntu Linux virtual machine that I described previously. I will be showing you how to use SAMOVA version 1.0. See the link in the show notes. SAMOVA 2.0 is the most recent version, but it does not work with Wine on Linux. And in any event, the main difference is that 2.0 has a bunch of undocumented or very poorly documented new features. So I prefer SAMOVA 1.0, which is simpler and more streamlined anyway. The first thing to do is open a web browser and navigate to my website, joshbanta.com. Next, click on Tutorials. Scroll down until you find How to do a Spatial Analysis of Molecular Variance. Click on Files Needed for Tutorial. Wait until everything is loaded on the page. Then click the Download button and select Direct Download. The download is complete. You can close the browser window. Open up File Explorer, which is pinned to the favorites on my virtual machine, or which you could find by using the search buttons below. Navigate to your Downloads folder and double-click on the file we just downloaded. The file we downloaded is in a compressed zip archive. This software is a compressed zip archive manager. We are able to preview the file within the Archive Manager software, but in order to uncompress it and have it available for use, we need to drag this folder out of the file compression software and into another folder. Go back to File Explorer, navigate to your home directory, and if you're on my virtual machine, there's a folder in there called bin. Double-click on that bin folder. And if you scroll down in here, you'll see that I already have a couple of folders called SAMOVA. These are from previous analyses running SAMOVA. What I recommend that you do is right-click if you're on a Windows-style computer with a Windows-style mouse, or if you're on a Mac laptop, push down on the trackpad with 2 fingers and select Rename. You can rename them anything you want. I'm renaming them with my initials and the number 1. And then we'll do the same to the other SAMOVA folder we have in here. Click Rename. Why did I do this? And why are there 2 separate SAMOVA folders in here anyway? Every time you run SAMOVA, if you follow this tutorial, you'll be putting a new SAMOVA folder into the bin directory. I don't want you to accidentally override the results from previous times where you've run SAMOVA for some other analysis of yours. Therefore, it's a good practice to just every time you run SAMOVA, to do it in a fresh new folder, and when you're done, rename that folder so that you can permanently save the results from when you ran SAMOVA on that one particular analysis. It has another benefit that if you ever need to come back and rerun the analysis for that particular data set you were working with, you can just go into that specific folder and rerun SAMOVA for that particular data set without influencing any of the other of your results and without overriding any of your other results. All right, now we will come back here and we'll go back to the home directory. Make sure that we can see the bin folder. So we're not in the bin folder anymore, but we can see it. Now we come back to our archive management software, and we'll take this SAMOVA folder that's in the archive manager. We'll drag it to the bin folder. This copies the SAMOVA folder out of the compressed archive and uncompresses it so that it's available for use within our bin directory. And there you can see the folder. You can now close the archive manager. Let's double click on the folder we just downloaded and have a look at one of the files. Double click on this file, 25 pops, SAMOVA, etc., This is the data we will be using for today's tutorial. If you're using your own data, you will have some other file ending in .arp that you will be putting into this folder for you to analyze. So remember that when going through this tutorial, anytime where I'm talking about this file called 25 pops underscore SAMOVA 2 underscore 1 underscore 001.arp, if you're using your own data that has your own file name, just remember that you'll need to take that file, put it in this folder, and all of the instructions that I'm giving you here, you'll need to change those instructions to work with the name of your file, although it will end in .arp. This .arp file extension is the Harlequin file format. I have a tutorial showing you how to turn your genetic data into the Harlequin file format. See the show notes for a link to my tutorial, how to make an Harlequin file from a FASTA file, if need be. Or if you're just following along with the tutorial today, you can just use this tutorial data. I will double click on the file, and it opens it up with text editor. Assuming that you made your Harlequin file using my previous tutorial, how to make an Harlequin file from a FASTA file, you will have some extra data on the end that looks like this. So I've scrolled down using the scroll bar to the bottom of the file. And there's this part here that says structure name, number of groups, populations, number of groups, etc. All of this has to be deleted for the file to work with SAMOVA. This particular part of the Harlequin file is useful for other analyses, but it does not work with the SAMOVA program, which, although the SAMOVA program is related to Harlequin and written by the same person, this particular part of the Harlequin file cannot be used by SAMOVA. But this data here is actually useful for us because it contains the names of all of our samples. So we're going to do something. We're going to delete it, but we're going to delete it by cutting it. So highlight everything as I've shown. Come here to the beginning of line 404 and get all the way through line 428. And then when you're somewhere in the highlighted area on a Windows style mouse, you're going to right click. On a Mac laptop, you're going to push down on the trackpad with 2 fingers and you're going to select cut. Now the rest of this information here, structure name, etc., and those 2 brackets, we don't need any of that. So we're going to highlight it and we're going to delete it. We also don't need lines 398 or 399, so we're going to highlight them and delete it. Now we will click on Save. And now we will close the Harlequin file. Because this is a spatial analysis of variance, we also need to provide spatial data. I will show you how to do that. Open up LibreOffice Calc, which I have pinned to my favorites or which you can find by searching. If you have old data that's still within the software, you can click on Discard. and you can click on yes for are you sure? And then you can click on cancel. Next, come to cell B1, click on it. Then, if you're on a Windows-style computer with a Windows-style mouse, you'll right-click. On a Mac laptop, you'll push down with 2 fingers on the trackpad and click on Paste. In this dialog window, all of the defaults look fine, so click on OK. We have an extra column here that we don't need that appeared, so I'm going to click on this cell here that says B, and that highlights the whole column. Windows-style mouse, right-click, Mac laptop, push down on the trackpad with 2 fingers. We're going to click on Delete Columns. Now, in this tutorial, the names of my individuals are sample 1, sample 2, sample 3, etc. These are the individuals for which we have genetic data and for which we have spatial data. I'll describe the spatial data momentarily. With your actual data, your individuals have some name that's whatever you assigned them when you originally set up your genetic data. So yours will not be called sample 1, sample 2, sample 3, etc. If you did what I showed you, where you cut this data out from the Harlequin file and pasted it into here, then these names will reflect the names of whatever you called your individuals in your study. However, if you're entering this in manually, make sure that whatever names you use here match exactly the names that are used in your Harlequin file. There can't be any differences. For instance, there can't be any spaces at the ends of the names, because that would be interpreted as a different name. And these would need to appear in the same order as the order in which your individuals appear in the Harlequin file. Again, these are each individuals, and these are the names of these individuals for which we have genetic sequence data in the form of DNA, and for which we have spatial data. The next thing we need to do is have a column of numbers. I don't know why the SAMOVA format requires the file to have this sequence of numbers in the far left column, but it does. So we'll just simply go ahead and do it. So here's the fastest way to do it. I'll type in the number 1, and then underneath it, I'll type in the number 2. LibreOffice will recognize this as a pattern. So if I highlight 1 and 2, and then if I get the little crosshair to appear by going to the corner of that second box, I can drag all the way down to row 25. And LibreOffice figured out that I was trying to write numbers in a sequence. So however many individuals you have in the left-hand column, you need to have a sequence of numbers that counts through all of your individuals, such as you see here. Now, in these two columns, this is where we put our GPS coordinates. GPS coordinates refers to the specific location in the world. So imagine if we took the globe and we flattened it out into a piece of paper, and we turned that piece of paper into graph paper. You could count along on the x-axis to a particular spot, and you could count along on the y-axis to a particular spot. And this would allow you to find any unique location on the Earth using two numbers. One number corresponding to the x-axis, the longitude, and one number corresponding to the y-axis, the latitude. You need to have this information for your own data if you're bringing your own data to this tutorial. So you have to have collected that data or have been provided with that GPS information for each of your individuals in the study. The GPS data has to be in the form of the WGS 1984 pseudo projection. This is the typical format of GPS data if the GPS data is coming from a GPS app on your phone or a GPS unit that you buy for the purpose of recording GPS coordinates. Another important point, that WGS 1984 data has to be in the form of decimal degrees. It cannot be in the form of degrees, minutes, seconds. If you're bringing your own data to this tutorial and your GPS data is in the form of WGS 1984 degrees, minutes, seconds, there are plenty of websites online that will allow you to convert from degrees, minutes, seconds into decimal degrees. So you'll need to make those conversions before you put the GPS data into this file. The next step is to add the GPS data as I'm showing you here on your screen. So you'll need to pause this video and go through and type in this GPS data as you see it into your file. Now, the important thing to note here is that the first column refers to longitude. The second column refers to latitude. So when you enter in your GPS data, you're going to do it just as you see here if you're just following along with the tutorial data. But if you're bringing your own data to this tutorial, make sure that in the first column you put longitude and in the second column you put latitude. It's very important not to get those flipped. Finally, we need to have a column at the end of just the number 1. Why the computer software needs that, I don't really know, but it's what we have to do as part of the file format. So what we can do is enter the number 1 3 times so that it's enough for LibreOffice to recognize a pattern. You can come down here into the corner till you get this crosshairs, and then you can drag that down all the way to the bottom and it will fill it in with the number 1. There's one thing we need to do to get this spatial data companion file in the right format. SAMOVA is very picky that the names of your individuals have to be in quotation marks. Here is a quick way to do that. Come to cell F1, type $, then type CHAR, which stands for character, and we're going to want character 34, which is a quotation mark symbol. Then put a space and the AND symbol, then another space. Click on cell B1 where we have the name of our individual. Then put in another space, another AND symbol, another space, type CHAR again, and parentheses 34 again. So this takes what's in B1 and puts it in quotation marks. This is a time saver because now what we can do is move our mouse to the bottom corner until we get the crosshairs, and we can drag that down for all of them. We're still not quite there, but this gets us closer. Now, with everything highlighted from the first sample to the 25th sample, I'm going to click somewhere in the middle here, where keeping everything highlighted. I'm going to well, not click because it'll do that. You need to highlight everything, come in the middle, and then right-click if you're on a Windows mouse, or push down with 2 fingers on the trackpad of your Mac laptop, and we're going to type copy. Now, come here to cell B1 where we have our first individual. Now, within that cell, right-click on a Windows mouse, push down with 2 fingers on a Mac trackpad. We're going to come down here to Paste Special, and then we're going to choose from the drop-down menu again, Paste Special. And from this menu, we're going to click OK. You are pasting data into cells that already contain data. This is fine because we want to update this data with the quotation marks. So we're going to click on Yes. There, now we've got the names of our individuals surrounded by quotation marks, and we don't need this cell, we don't need any of this data anymore. This will mess up our format. So I'm going to click on the F so that I have the whole column highlighted. Right-click Windows mouse, push down with 2 fingers on a Mac trackpad. select Delete Columns. We now have our data in the format needed for the companion spatial data for SAMOVA. Now we have to save it the right way. We're going to go to File, Save a Copy. Now we're going to navigate to the folder where we're working with today, which is in our home directory, in the bin folder, in this folder right here. We're going to double click on it. This is to make sure we save it in the right place. This is important also. We need to change the file format. We're going to click on the little triangle here. I'm going to hover over the little triangle here to see all the choices. We're going to select Text CSV. We'll keep the file name as Untitled1 for now, although we are going to change it. Choose Save. Now in here, this is important. From the drop-down menu under Field Delimiter, you probably will have it showing, when you open up yours, you'll probably have it showing like this. From the drop-down menu under Field Delimiter, choose Tab. For the drop-down menu under String Delimiter, switch it from double quotes to single quotes and click OK. We now have the data properly saved, and we can close LibreOffice. Save changes to the document. Don't save. We already have the changes that we need. So next we're going to open up File Explorer. Navigate to our bin directory. We're going to go to the folder that we're working with today. And here we have this file called untitled1.csv. We have to change everything about the name of this file. This is the file that we just made in LibreOffice Calc, but the name is wrong. SAMOVA requires that your companion spatial data have the same name as your Harlequin file that contains your genetic data. If you're using my tutorial and you're going along and using my sample data, we need to make the companion data file have the same name as this one here. But if you are using your own actual data with this tutorial, you will need the name of your companion data to match the name of whatever the Harlequin file is with the .arp extension that you brought and that you put in this folder and that you're using. But assuming you want to just use the data that is part of this tutorial, here's how we can make the file names match. Highlight the Harlequin file, right-click on a Windows mouse, two-finger push down on the trackpad on a Mac, select Rename. We're not actually going to be renaming anything, but this allows us access to the file name where we can copy it. So we're going to highlight just this part of the file name, not the file extension, not .arp, but just this part of the file name here. And then hovering the mouse somewhere over that file name, right-click on a Windows-style mouse, push down with two fingers on a Mac trackpad. We're going to choose Copy, don't choose Cut. Okay, now we can just come back up here and we can just click on the original file again to get rid of that. And now scroll back down. This is our companion spatial data where we need to change the name of the file. Right-click on a Windows-style mouse, push down with two fingers on a Mac trackpad, choose Rename. It has just the name part highlighted here. We need to delete the whole thing. So I highlighted the whole thing and I'm going to delete it. Now I'm going to come in here, right-click Windows mouse, push down two fingers Mac trackpad, choose Paste. We're not quite done. So here is the name of the file so it'll match perfectly. But now we need to add the file extension .geo, all lowercase. Now we choose Rename. And now we are ready to actually run SAMOVA. To do this, open up a terminal window. I have it pinned to my favorites, or you can search for it down here below. We need to navigate to the folder where we keep our SAMOVA program. So type CD, space, come back to File Explorer, navigate to the Home directory, navigate into the bin directory by double-clicking on it. Now here's the folder we're working with today. Take this folder, drag it into the terminal window. Now come back to the terminal window and hit enter. Type wine, and now all caps, SAMOVA, all capital letters, dot, and then lowercase e, lowercase x, lowercase e, and press enter. And here is our dialog window for running SAMOVA. Generic name of input files. This is the name of your Harlequin data and your companion spatial data that accompanies the Harlequin genetic data. We'll do this with copy and paste again to make sure that we type in the name of the file exactly right, with no differences in this, in having an extra empty space or a lowercase where it should be an uppercase. It'll work smoother if we just do a copy and paste. So we'll come back here, we'll open up File Explorer again, navigate to the bin directory again. We'll use the folder that we're working with today. We'll come here to the Harlequin file, right-click on a Windows mouse, push down on the trackpad with 2 fingers on a Mac laptop, choose Rename. Having only this part of the file highlighted without the file extension, hover the mouse over that part somewhere, right-click on a Windows mouse, push down with 2 fingers on a Mac trackpad, choose Copy, not Cut. Okay, now we can just click anywhere on this file. Now, come back to the SAMOVA dialog window. If you can't find it, it might have gone underneath the terminal window, and you can move the terminal window to find it. Come back into here. Highlight this name that they have in there, which is not the name that we want. Delete it. Now, right-click on a Windows mouse, push down with 2 fingers on a Mac trackpad, and choose Paste. So now we have it exactly right. But notice there's no file extension here. That .arp at the end is not there. But besides that, we've got the name of the file, which is also the name of the companion spatial data file, written in there exactly correctly. This next setting here is the number of groups. We're going to keep the number of groups set to 2. This is how many distinct groups you want the SAMOVA program to find. How many distinct groups that are genetically distinct from each other and spatially distinct from each other do you want SAMOVA to find? Now you're wondering, I'm sure, well, how do I know how many groups I want SAMOVA to find? Because it's likely you don't know how many groups you really have in your data set. What you want to do in that situation is you're going to want to run SAMOVA like I'm showing you right here, and you're going to want to run it for over and over again. You're going to want to run it for 2 groups, for 3 groups, for 4 groups, for 5 groups, etc., probably up to around 10 groups. Or you could run it up to even more than 10 groups if you wanted to. Every time you run it, make sure that you don't override your results from the last time. So you'll probably want to put it all in a different folder or something like that. For this tutorial here today, we're only going to be running it for 2 groups. That's all you need to do. But for your own real data, recognize that you're going to want to rerun the SAMOVA over and over again for the number of groups equal to 2, number of groups equal to 3, 4, and so on, so that the SAMOVA will try to find the most genetically distinct 2 groups that are also spatially distinct. And then you'll run it again, so it'll try to find the most genetically distinct 3 groups that are also spatially distinct, and so on. And then what you would do if this was your real data, which you don't have to do for this tutorial, is you would then compare the results and find which set of results are the best ones to use. How would you know which set of results are the best ones to use? Well, I'll just tell you right now that typically what people do is they look for the results that have the highest FCT value. Not the highest p-value for FCT, but just the highest FCT value that is also significant. Those are just some details to be aware of if you're working with your own data. The next thing to be aware of here is this box that says number of initial conditions. We're going to set this to 10 so that it will run fast. If you were using your own data, you would probably want to set this to 100 or even higher. This is how many different ways SAMOVA will try to partition the data and then to see if the results are similar across the different runs, and it'll pick the best partitioning of the data as your final result. If you want to read more about the best practices for running SAMOVA with your actual real data, as opposed to the faster way I'm showing here, I have provided the link to 2 research papers that were published in the peer-reviewed literature that provide some good methods for how to actually run SAMOVA with your own data in terms of what settings to use. But to make it run fast for now, we'll set the number of initial conditions to 10. For the molecular distance, we have 2 choices, either DNA data or microsatellite data. Since this is based on DNA data, we will choose pairwise difference. And now we will select Run. We received an error code there that the program aborted. Here's what we can do to make it work. I'm not sure why we're getting that error code, but something that seems to help. If you go to File Explorer and you go to that .go file and double click on it, it'll open up in Text Editor. Verify that everything looks as it should. And that there's spaces and that the individual names are in quotation marks as they should be. Click on Save and close it again. Sometimes opening it in Text Editor, saving it, and closing it again fixes the .go file so that it will run properly. So let's see if it worked. So we'll type in again wine, SAMOVA, all uppercase SAMOVA.exe, all lowercase.exe. We'll come in here, we'll delete that, we'll put in the name of our file, set this to 2, we'll set this to 10, we'll run it again. There, now it ran successfully with no errors. And here's the results. Let's start by looking at the file SAMOVA results arlequin.txt. To find that, you'd go to File Explorer, you'd go to Home, you'd go to the bin folder, you'd go to the SAMOVA folder that we're working with, and you'd look for this file SAMOVA results arlequin.txt. Depending on how you have your data sorted, you may have to scroll down to find it. Double click on it. And here's our results. Here's an AMOVA table. This is done using an analysis of molecular variance in the program Arlequin. I have a tutorial about how to use the program Arlequin. See the show notes below. The results that we want to look for are FCT, and we want to look at the p-value down here below for FCT. A p-value below 0.05 is considered significant, so this is considered marginally significant. FCT is a measure of how different genetically the 2 populations are from one another, because remember that we told Arlequin to find 2 different populations that were spatially and genetically separated from each other. And then this analysis of molecular variance tells us how genetically separate the 2 populations are from one another, how genetically distinct they are from one another. This number ranges from 0 to 1, with 0 being that the 2 populations are not genetically differentiated from each other, and 1 being that the 2 populations are completely genetically differentiated from one another. So this is kind of somewhere in the middle. So those are the 2 numbers that you would report, the FCT and the p-value. But there's also a whole AMOVA table here if you wanted it. Next, let's look at the results SAMOVA final structure.arp. Double click on it. And this will show us, if we scroll down, this looks like the input Arlequin file that you originally put into Arlequin. But if you look down below, it's put in this stuff, definition of the group structure. Remember, we took this out when we ran it, but now it's back, and now SAMOVA has put the individuals into two different groups depending upon which group they're in genetically and spatially that's distinct from the other group. So SAMOVA found one group that consisted of all of the samples except for sample 1. And SAMOVA found that sample 1 was genetically and spatially distinct and put it in its own separate group. If you told SAMOVA to look for 4 groups, you would have the individuals divided up into 4 groups down here below instead of 2. Using that information, you could then make a figure like this one. I have a link to this journal article in the show notes, where you would take your individuals, show where they occur on a map, and you could color code them according to what population they belong to as identified by SAMOVA. Now, to do this would require using Geographic Information System software, GIS software, which is beyond the scope of this tutorial to make a figure like this. But I do have tutorials about using GIS software, including how to make publication-ready figures on my website. See the show notes for details. So there you go. That is how you use SAMOVA.